Hi everyone. This is just a quick walkthrough of lab exercise 10 to get you oriented and ready to do the last exercise of Bio 139 lab. This is the urinary physiology. Hopefully everyone has already gone through the urinary anatomy lab that was coupled with the respiratory physiology and the respiratory anatomy and the digestive anatomy. And also, you are very likely in the urinary physiology in lecture by this point. If not, you should be getting pretty close to it. So today, what we're going to be learning is how different aspects of our diet affect the function of our kidneys and how they will alter the production of urine based on that. So on the cover page of our lab, we have, as always, the purpose to study the urinalysis and interpret varying results. So we are going to, as part of your virtual lab activity, you are going to perform a virtual urinalysis. Here are your learning objectives, your lab activities, and then of course, the different parts of the lab, including some case studies, which we will not go over, but as you work on them, as always, you can reach out to me with any questions or just to get some feedback on what you think the correct answers are. So, background, the renal system. Renal just means pertaining to the kidneys. So, renal system, urinary system, they're used interchangeably. The terms that you will see a lot, especially in lecture, but also in lab: filtration, reabsorption, secretion, and excretion. And those are words that we've heard before, but when we use them pertaining to the kidneys and the urinary system, they actually mean something very, very specific. So filtration is the first process of taking substances out of the blood, ultimately to make urine. But let's be a little bit more specific. In the kidneys, there are small units called nephrons, and nephrons are the functional unit of the kidney. There are roughly 1 million of them in each kidney. And as blood travels to one of these nephrons, there's a special capillary called a glomerulus. And I like to describe a glomerulus as a watered up garden hose that someone has poked holes in. So as the blood travels through this glomerulus, it's like water traveling through that garden hose. Liquid is going to spray out, and it's spraying out under pressure. But now imagine if that water traveling through the garden hose had particles in it. Some of those particles may come out with the water. Well, as the blood travels through the glomerulus, some things that are in that blood are going to come out and some are not. Some things are going to come out because they're really, really small, but there's also things like charge that plays a role. But that's really more of a topic for lecture. What we need to know for lab is just that filtration is the process that occurs at the glomerulus. And it is taking substances in the blood and removing them to place them into the nephron. And it happens under pressure. So it is a passive process. Now, when we do this, the substance that's produced, that liquid that's being collected inside the nephron, this is called filtrate. It's very similar to plasma. It's not identical to plasma, but it's very similar to plasma. And it's also not yet urine. It's similar to urine, but it's not urine. It still has a lot to happen to it before it becomes urine. So that first little bit of the nephron, there's this shell that surrounds the glomerulus called Bowman's capsule. So Bowman's capsule is filling up with this filtrate. and the filtrate now moves into the rest of the nephron, this long series of tubules. And as it moves through the tubules, the next two processes occur: reabsorption and secretion. Well, that filtrate, it's mostly water, but it's got a lot of things like ions, nutrients, things like vitamins and amino acids, glucose, all sorts of stuff dissolved in it. And a lot of that stuff is things that our body really wants to hold on to. So reabsorption is how our body reclaims the things that it does want to hold on to. So a way that we can describe reabsorption is the process of moving substances from the filtrate back into the blood along the tubules of the nephron. Secretion is kind of the opposite of reabsorption. Secretion is the act of moving substances from the blood into the filtrate along the tubules of the nephron. Well, why would we need to do that if we already filtered? Well, remember what I said? Some things were too big to be filtered, or some things were not filtered because they had a certain charge. Well, things that were not filtered, but our body does need to get rid of, they are secreted. They are moved from capillaries that surround the nephron into the tubules of the nephron. Now, by the time we get to the end of all of those tubules, we have reabsorbed most of that filtrate. We have secreted a little bit of stuff into that filtrate. And that filtrate at the end of the tubules is now urine. That urine leaves the kidneys, it travels through the ureters, and it is held in the urinary bladder until we get to the toilet. And that is where excretion happens. Excretion is as the urine leaves our body. And the act of urination is also called micturition. Micturition and urination are forms of excretion. All right, so the rest of this paragraph is just kind of reviewing topics that we've talked about previously in Bio 139 and 137: osmolarity, solution, solutes, solvent. Make sure and review those if they're a little hazy for you. But let's get down to talk about a urinalysis. What we see here is a diagram of a urinalysis test strip. You might have done one of these at a doctor's visit if you ever had to do a lab. Also, these, you can get them at many pharmacies. What happens is you will place some urine on each of these little squares, and there's a tube that comes with this, and you can hold these squares next to a little readout on that tube, and based on the color that these squares turn, it tells you the glucose level of your urine, the protein level of your urine, the pH, nitrate, leukocytes, esterase, ketones, bilirubin, urobilinogen, blood, and specific gravity of the urine. So it tells you on this one test strip quite a bit about your urine, and you will do this in your lab activity, your virtual lab activity. Now, in the pathophysiology section, we've talked about diabetes insipidus back in endocrine. Remember diabetes insipidus, there was central diabetes insipidus where the pituitary gland, specifically the posterior pituitary gland, either does not produce any ADH or it does not produce adequate levels of ADH. So what happens is your kidneys do not get the signal to reabsorb more water. And then there is another type of another type of diabetes insipidus called nephrogenic diabetes insipidus, in which case your pituitary gland does make ADH. It's just your kidneys don't respond to it. So here are some information about what we might find on the urinalysis. Normal results versus some things that might be abnormal results. Now, when we get abnormal results, there could be a couple of different reasons. There are pathological causes. These are causes that come from some sort of illness, maybe a disease or disorder of some sort. There are non-pathological causes, things that are not a disease or disorder, such as pregnancy. Pregnancy can cause high protein in the urine. That's actually how pregnancy tests work. Pregnancy tests test for very specific proteins in the urine. We wouldn't call that pathological. That's not a disease or a disorder. So it's considered a non-pathological cause of protein in the urine. So here are non-pathological causes of certain things in the urine versus pathological causes of things in the urine, as well as what the terms are. If we find protein in the urine, the term for that is proteinuria. If we find glucose in the urine, it's glycosuria or glucoseuria. Ketones in the urine is ketonuria. So for the most part, now not always, but for the most part, the term for finding something in the urine is just whatever it is with urea at the end of it. Now that's not always the case, but a lot of the time it is. Here are some virtual lab activities that you will click on to get to those virtual labs. And like I said, a couple of case studies. Make sure to do those case studies so that you can practice them before your last lab practical of the semester. If you have any questions, please reach out about them. I will not give you the answers, but I'm happy to give you feedback on the answers. All right. Take care and good luck on your last lab exam of the semester coming very, very soon.